an odds ratio of 1.5 for breast and an odds ratio of 2.1. And we were very skeptical because epidemiology has really failed with prostate cancer. We haven't really identified risk factors for prostate, apart from the genetics that we can explain a big chunk of. But obviously there are environmental factors, otherwise you wouldn't have trends in prostate cancer incidence if there were not environmental factors. And we haven't found them. So this, made us be very cautious about our results. It got a lot of publicity. It was published a few months ago in the HP, and it got a lot of publicity, media coverage, and we were very, very careful about what we found. We didn't want to overblow our findings. We tried to see what bias could have produced it, and we couldn't find. We adjusted for whatever we know, but for a disease that you don't know the risk factors, you cannot adjust much. I mean, we did adjust, but it was not even necessary to adjust, and probably. And we were particularly concerned about socioeconomic status, so we adjusted for the educational level of the subject, for a score that takes into account the socioeconomic status of the parents of the subject, and urban index, which is an area-based SES score. And so that's what we're finding. And we are now, we want to see whether other people can replicate, and we are now moving to much more detailed exposure assessment to see whether what we have in front of the house, the light you have in front of the house, how important it is and how it coincides with light inside the house, and how much it coincides actually with measured light outside the house. So now that's what we're doing with studies with the wearables. And we had a long discussion with the reviewers because they were saying you should measure inside the house. Well, we hadn't measured inside the house. We had a Likert scale, a questionnaire for inside the house, where we were asking how bright it is when you sleep, et cetera, and it gave the same results. But it's a Likert scale. It's much. So this is better, and this is Spain. So we are much more outside than what probably you are here. So. And what we were arguing also is that the time before you sleep is important. And that's what, as far as we understand more light, this is what we're getting. It's not only the time when you sleep. If anybody wants to comment during the talk, please do. So the summary from the blue light paper is that we are trying to find an association of light at night exposure with hormone-related cancers, breast and prostate. And this is among the few studies that have individual, actually, data as we did it, and that applied the new method. So, and I think the nice bit of this paper is that we stress the importance to look at light spectra, not on the irradiance, based on the overall. So I think that is the nicest part of the paper. And obviously for public health, if what we are saying is true, I'm not sure it's true, but if it is true, it is important because we clearly have much more exposure to blue light spectra because our cities are converting to LEDs, and they are converting to the hard LEDs, the white ones, not the red ones. And now what we are doing is we are doing a number of exposure assessment studies, and it would be great if other people did it also. So let me move to, yeah.
So before you leave that topic, it seems to me, could that be a confounder of socioeconomics, that if you have richer areas, they can afford the white light LEDs, and maybe more rural areas don't?
Yeah, these are all urban areas, and there are huge socioeconomic differences within Barcelona and within Madrid, very, very big. And we went back to get the images as back as we could. And definitely before the cities switched to LEDs. So this is not LEDs, not massive LEDs at least. So we are capturing light before the global change. There were some changes. I think that there are a couple of problems here. First, that the satellites are not capturing very well intensity. The light is not just irradiated out, up in a way that is necessarily what we are getting. And we are modeling proportions here, a proportion of blue light, not the cumulative, so it's not multiplied by intensity. And we may have socioeconomic factors, and we control for SES as good as we could, and we had the area-based SES, and it didn't explain it. But, I do take your point. It could be some other factor in SES related that, we just couldn't adjust. But as far as we adjusted, it just didn't change too much. It changed a bit. So, but it could be something else. And I think, for prostate cancer, really, you have to be very careful. So we were very careful in our interpretation. But I thought it was very good to publish because it was just new methods. And for prostate, if you don't have good hypotheses, it's good to get some new ideas, in fact. So let me move into timing of diet, which is related to the graph I showed for the mice before. So at some point in time, I was new in the field. We had done a study, so we recontacted 10,000 people. We called everybody again, and we asked them specific questions on the timing of diet and of physical activity. And I thought that was important because clearly that's what the animal studies were showing us. And this is a really nice paper in PNAS on evolution of diet, and basically what we wanted to compare is different patterns in terms of timing of diet. And all our big cohort studies, none of the big cohort studies, nurses, EPIC, have looked at timing. We are looking at what we eat, veggies, red meat, processed meat, how much we eat, and we're not looking when we eat. And the animal studies indicate that when we eat is also important. And you could have different patterns. You could have 3 small meals during the day, or as many of us have in our societies, 3 big meals and 2 others, and some of it during the night. Or you can have nothing, which is very good. So, and that's what we asked. We contacted everybody again, and we asked them about their patterns. When did they eat, and whether they were snacking, and when did they take the snacks, and all that stuff. And that's what we find for breast and for prostate. So this is time, and I'll show you some more data in a minute, time between your last big meal, dinner, and the time you sleep. Based on the assumption that if you eat, and we adjust for quality of food and quantity here. If you eat and you go to sleep, you're doing it wrong on the basis of what we saw from the, and you don't need an epidemiologist to tell you that you're doing it wrong for the immediate effects. You don't sleep well. If you eat a lot and you just go to sleep and you drink and you go to sleep, you just don't sleep well. You don't need a study, it. But we are looking at long-term effects here. Does it have long-term effects also? And we are finding a protection for people who space more the time between dinner and sleep, which I thought was interesting. And we published it a few months ago, and again, it got a lot of media coverage. And we had put the time at 2 hours, as that's the stratified analysis we did, but I'm not suggesting that it's necessarily 2 hours. I'm just saying that this is an interval that you should take into account in your studies. And the other thing we modeled is not only dinner-sleep interval, but time of dinner. And it's basically these 2 variables we cannot distinguish. They are the same. They are very correlated. And median time of dinner in this population is 9 p.m. in Spain. So 50% have dinner before and 50% after. And if you look at the odds ratios for breast and prostate, and if you combine them, it's about, whether you do this or whether you look at that, is about 20% decreased risk. So you're protected if you eat early, you're protected if you leave time before your dinner and you sleep, and if you do both, it goes to about 25% protection, because they're correlated. So it's just a bit more protection. And yeah, people say 9 p.m. Well, that's not really early, but this is Spain. So Spain also is below London. We have the wrong time. So in terms of the position, light, it's 8 p.m. So it is not as late. But still, for here, it is late. But that's how it is. That's what we find. And I'll show you the findings of another study that did exactly the same, was published the same month, and they found the same. They are the only two studies we have on timing in cancer. And when we talk, for example, about the US or the Nordic countries, and it will be great to get some more studies in different social settings, people say, okay, here we eat at 7. And you talk to chronobiologists, and they would say, yeah, we eat at 7, but then we have something to eat before we sleep, a yogurt or whatever. And you talk to guys who are in chronobiology, and they say, that's not good, because even if you eat a yogurt, you just restart the whole system. Your liver starts functioning again. Well, the time of repose between dinner and the time you wake up, this is messed up. You eat at 7, but then at 12 o'clock or 11 o'clock, before you go to sleep, you eat a yogurt, you restart the whole system. Whether this is true or not, we don't have epi data on that. And this is what the French, and they published more or less the same time as us. And this is, and they expressed it the other way around. We expressed it as protection, and they expressed it as risk, but it's the same thing. And they found they didn't look at the interval; they just look at time. And it's France, so they eat a bit earlier, so they have only 10% eating after having dinner after 9:30, and they find the same basically. And these are the only two studies we have. And it's really a pity that the big studies that on diet, on nutrition, have not looked at that, and I think they should be looking at that. And actually, we are now trying to replicate with a number of other studies. But basically, what we are finding, it would be nice to try to check in humans what was found in animals. And in animals, it indicates that timing is important. And I'm not saying it's exactly 9 p.m. or less, but it's timing that's important, and we should evaluate it. Let's move a bit on mechanisms. These are studies we have done in the Volkswagen factory, the SEAT factory in Barcelona, and they have about 14,000 workers in the plant in the south of Barcelona, and they have 9,000 workers rotating, and they rotate the way you see: 3 weeks night, 3 weeks evening, 3 weeks morning, and it's like an experiment basically, because after 3 weeks continuous night, you are entirely adapted to night. It's like when you go for jet lag, more or less 1 hour per day you adapt. So you stay 1 week or 2 weeks in Europe and you're adapted, more or less, not entirely, but more or less. So we got the same people. So it's a within subject comparison at the end of the 3 weeks night when they were adapted to night, and then at the end of the 3 weeks morning. And obviously we did it in a random way. Some people entered with the morning and some people entered with the night. So they didn't all of them start at night. And we got blood samples before and after work. So we have 4 blood samples per subject. And we got all the urine voids when we sampled during 24 hours. And we did a number of other stuff, measured light and sleep and physical activity and electromagnetic fields. And we basically are showing, we are now analyzing, but what we are finding is that it's more than melatonin. That's what we are finding. So, the light, this is from a pilot study, the light type of data we would get, and unfortunately we didn't have enough money to get a wearable that would get us the spectrum. So it's overall light, which is a pity. So a day worker, would have exposure to light and then would sleep, so no light, and then light again. And a night worker, this is in nurses, this is not from the state, it's from a pilot study. So they would be in the hospital, dim light, go home, lots of light, sleep, no light, light again. And this dim light that we have unless you are in operating room, but in the wards, is enough to suppress your melatonin. You don't need much light to suppress melatonin. So even that would suppress melatonin. So we have this type of information, and we have all the urine voids, and we have analyzed for sex hormones and melatonin, and that's the type of thing you would get from each worker. This is from the day shift. And you see that you can plot the day-night pattern, and just seeing very, very superficially, you can see that most people follow this pattern. Not all. I mean, this person does not follow, this person doesn't follow well, but most people, would follow when you plot their melatonin levels in all the urine voids of 24 hours, they would follow this pattern. And then you combine and you compare the same people when they were doing night or whether they were doing day, so we don't have confounding, basically, here. And what we find is, this is the curve for the people when they were doing the day, this is when they were doing the night. And if you look at the area under the curve, it is very similar. So they have a bit less of melatonin when they're doing night, but not much, which means they're adapted. But look at the time when the melatonin peaks. For the night worker, it peaks sometime early morning, and when they do the day, when they do the day, it peaks as it should during the night. And when they do the night shift, it peaks at about between 1 and 2 p.m. So huge difference, really huge. So they get enough melatonin, they get it the wrong time. And I think that's important for a number of variables. You may be getting, what you get even if you don't work at night, but you get it the wrong time. And that's where we are. And we looked at the and so this is what we would have, for example, if we measured, so our melatonin will go up at night. If we party one night, so we sleep in the morning, our melatonin would still peak during the night. If we party for a whole week, our melatonin will move the peak. And that's what happens with them, because there are 3 weeks continuously night, 3 weeks continuously evening, 3 weeks continuously. So they're adapted. And then we look at the sex steroids. And this is more or less the same analysis. It is night shift versus day shift. What time do the sex steroid hormones peak? And we had a number of, quite a few of them. This is cortisol. You cannot see the names, but this is cortisol and other corticosteroids. This is the group of androgens. This is progestogen. This is estrogen. These are all men. And this is the time that the hormones peak as compared to in the night as compared to the time they do day. And you can see, for example, cortisol. You can see all the androgens. You can see progestogen. So, and I think this is a really interesting pathway if you're talking about breast and prostate cancer. Actually, you have changes in the time of peak for sex steroids that are clearly of interest for hormone-related outcomes. So, and I definitely think we have to be looking at other outcomes. And we now have analyzed the immune response. We have done a panel of cytokines and growth factors and chemokines. And we again have, and we haven't finished. I'm not showing it because we haven't finished this analysis. And again, we have some very interesting pathways modified. And we are not, and we finish also the RNA-seq analysis, the gene transcripts, and we don't get as much differences as I would expect, in fact. Much less in terms of gene expression than what I would expect. So we have clear effects on phase shift, later peak of melatonin, but not clear suppression. We have changes in phase shift and production. I didn't show it of selected sex steroids, and I haven't shown the immune response. So very, very sketchy comments on prevention. I'm sorry. The first thing is, what do we do with sleep? I mean, sleep is a factor which is important. And we had last month this really nice paper coming in from Seattle, from the group of De La Iglesia, who is a chronobiologist at the UW. And they took advantage of the change that was done in the School of Seattle for the time of entrance of the high schoolers. They were entering at 7:50, and then it changed because there was a perception that they were not having enough sleep, and they are entering now at 1 hour later, practically 50 minutes later. So 8:50 instead of 8:45 instead of 7:50. And then some people said, okay, they will just stay late at night. If we ask them to wake up later, they will just stay late at night. They will not gain in terms of sleep. And Horacio de la Iglesia happened to have quite a few actigraphs available, so he just managed to get his team to mobilize very quickly and got 2 schools, and he put actigraphs to about 400 high schoolers before the change and after the change. And he showed that, first of all, physical activity, the blue is after the change. Physical activity starts late, and this is the class, no class, or whatever, but it starts later. And look at the duration. They do sleep a bit later, but they basically sleep 35 minutes more. So it's important because we know that adolescents have a lack of sleep from many studies. So one way of prevention, which is sleep. Another big chapter in prevention is light. That's what we were talking, Laurel, before. And this is the type of thing that has been done in a number of hospitals where they have switched to this type of very bright light, which is entirely unnecessary. You have the patients there, and they wake up at 3 o'clock in the morning, and they have this bright light coming to them, and they wake them up, and they suppress them a lot. And you don't need it. You're not operating. They're just sleeping. So there's no point in having this type of thing. And they are moving to this type of pattern, which is good enough. You're not operating there. For an industry, it might not be good enough because then you may increase the accidents and the sleepiness. But for a hospital, it's good enough. Yeah.
Yeah, this is very interesting. But blue light and bright light is good in the morning. We need that.
In the morning, yeah.
In the morning. So
absolutely.
And the industry is moving already to lights that are tunable. They tune
exactly.
to the natural sun cycle.
Exactly.
So the question, this is the right time to do experimental studies with tunable lights that already exist, that are bluer in the morning and softer and reddish in the evening.
I agree, absolutely.
There, cancer will take, along
yeah.
blue.
You do intermediate markers, you have to look. Sure. I agree, absolutely. That's one of my conclusions, that we don't have, we don't have these studies. And we're doing a lot of interventions without evaluating them, in fact. I agree, absolutely. And this is an example of a place where they actually have this type of lights, and they change from bright to red. And the other thing is to try to do personal prevention. And you would talk to a chronobiologist about, say, a nurse who rotates, who does 2 nights one week and then 3 nights the other week, etc., and say, fix it so that this person sleeps well. And they can more or less fix it, but it's a very complicated chart what they give you, like this one. I'm not going into detail. So you have to do different things in different days. You have to get melatonin 1 hour, and then the next day another hour, and you have to get light baths, and you have to do and also. And for those of us who have worked in occupational health, and they manage to get the lowest temperature at the time you're sleeping. This is the idea. The lowest temperature of the body is about 4 o'clock in the morning. That's what they try to do, to make sure that you sleep. That's indication of you sleep well. But you go to a factory with this scheme, and there will be a few workers who can do it. They are really concerned, but prevention just doesn't work with so complex schemes. It doesn't work. And so we have to do something else, and we haven't found it. And I'm supposedly advising my government, and I don't know what I'm going to tell them. So circadian disruption and night shift work associated with acute and chronic health effects. They have been associated with cancer and cardiometabolic. We have really, I think, a problem with our evidence that we haven't looked at the general population on issues like light at night and mistimed activities of the general population. And clearly what Javier was saying, that we really need to do some more evaluation of the interventions. There are some that are obvious, but clearly we have to do evaluation of the recommendations. This is the immediate team working. This is my center. So here, and Ariana is postdoc, Michelle is assistant professor, and we have been doing a lot of studies together. Gemma is a project manager, Anna is a biostatistician, fantastic. And Kyriaki was a PhD, now she's assistant in the University of Vienna, one of the best PhD students I had, and we're working with a lot of people in the labs. So, thanks a lot.
How much research has been done on circumpolar populations of people who experience really dramatic, pure night, pure day, and then everything in between, but they've evolved under that. It seems like maybe there might be some insight by looking at animals.
I think so. There has been ecological studies that have looked different populations of the world, and they find associations with light and breast cancer and prostate cancer. The problem with these ecological studies is that apart from the light, the Swedes are different in many ways from the Spaniards in many other ways, so it's not clear that it's light that produces the difference as compared to other things. And we don't have good studies with actigraphs and light measurements in this very special population. I think that would be a fantastic population to look at. I agree. Yeah.
Thank you. Now, you looked at diabetes and myocardial infarction. Don't you think that it is more associated with stress of night workers than the shift work itself?
It could be. It could be, and it could be very also clearly associated with sleep disturbances. But unless we measure it with one way or another, and this study I showed on the car factory, it's good because it's a nice design, but it's small. We have 50 workers, and we should have gone much more. And there, because we have a number of questionnaires. We have the STI for anxiety, for example, and a number of others, but we are small. We really have to get bigger studies and very well controlled, I think. I agree, absolutely.
We did that in the shows housing.
Yeah.
And we found an interaction synergy between shift work and resting diabetes and weight. That was especially stronger in shift workers who also have insufficient sleep.
Yeah.
But we did control for markers of psychosocial distress. We really found that was.
Yes.
Thank you for your wonderful presentation. How did you control for temporal issues of, cancers like breast cancer, prostate cancer that might take 10, 30 years to develop, and the exposure of light in a few of the studies? Or I'm just trying to wrap my head around it. I don't know how you.
Yeah. Well, we have a lifetime occupational history, so we can figure out the temporal relationship as concerning shift work, and that's how we actually evaluated duration of employment and time since first exposure and time since last exposure. So this we can do, and people remember their occupation. They may not remember exactly, the time they entered, so this is more tricky. And for many of the studies, it has been really tricky to, people say, I'm a day worker, and then you ask them what time you were working, and they were working for 4 o'clock in the morning, and then you say, 4 o'clock in the morning, you have messed up your circadian disruption. So we reclassify these people, even though they say, so we had detailed history. There is exposure misclassification. I doubt that it's differential, frankly speaking. And then for the light, we can go as back as we can when, from the availability of the pictures from the International Space Station. The good thing is that the lights of the cities are not changing so quickly. We have a big change the last 10 years to the LEDs, but before that, we have records for Barcelona and Madrid, and we know that lights have not changed for quite a long period. So I know we were evaluating a very recent period. This is not the time we would like to be evaluating. But probably it reflects quite well what happened at least 10 years before that. And we consciously got the pictures before they switched to LEDs, massive switch to LEDs in these two towns. That's how we manage. It's not optimal, but we do manage to look at some of these variables. Okay, Perry?
Thanks, that was super interesting. The prostate results are huge. In terms of, we don't know really any other non-genetic risk factor that could even confound those results.
Well, David found something different for prostate in your distress study, but you don't believe it very much. Yeah, I'm very cautious with my results also. It's similar as you.
Is screening a big issue in prostate cancer screening?
Yeah, prostate cancer screening, sure, but prostate cancer screening does not explain the increase in prostate cancer incidence. So there is something happening with prostate cancer, and we definitely are not looking at the correct risk factors, otherwise we would have found it, or we're not looking at the correct time of exposure. It could be that prostate cancer is similar to other diseases that you have, very early life exposures that are important, and we haven't looked at that, and we are not looking at that obviously here. But I think it wouldn't be bad for prostate cancer to get some new ideas out and check them, because we are really stuck with, prostate is among the 4 most common cancer in the world. And we only know about basically genetics and prostate cancer. And it's an important component, but it doesn't explain time trends. Genetics don't explain time trends.
So if I pushed you on the early life risk factors of prostate cancer with limited funding and whatnot, what would you think would be the candidate factors?
Well, it has to be hormonal, some way of finding either intrautero or early life or adolescence. We have tried to do it measuring anogenital distance in prostate cancer patients. Anogenital distance is the distance between the anus and the genitals. And it has been looked particularly for endocrine disrupting chemicals in children, phthalates and others. And it's very different between males and females, humans and animals. So vets actually look at this distance. Sometimes it's not clear whether puppy or whatever newborn animal is male or female. They look at this distance and they can define whether it's a male or female because it's longer in males as compared to females. So, and there was this suggestion that you could measure it and associate it with in babies, associated with endocrine disrupting chemicals, and they have found associations with a number of endocrine disrupting. So we did do a small study on prostate cancer cases and urological controls, and we found a difference. And now we are repeating with a bigger study. I'm not sure, if it's the best thing you can do because we don't know what determines anogenital distance during life. But we would have to find some of the really nice prospective cohorts that have, but we would have to have biological samples. It wouldn't work, I think, in other ways, and do some analysis there. That's what I would do if I had the samples.
Yeah, as I recall, David Barker found effects for birth weight and early growth.
Oh yeah, sure.
Those are the only ones that pop into my head.
There's huge literature on early effects. Oh, absolutely. They have big conferences every year, the DOHAT people, Developmental Origins of Health and Disease, for many different outcomes, actually.
Yeah, normally more orientated towards cardiovascular.
Yeah, sure. Okay. Yeah.
So what kind of research can be done at a local level?
That's good.
Like a local level, so like at our cancer center here, what can we do to, see what like across Oregon?
Oh, there are lots of things to be done, and there's a lot, I think, to be done also in terms of interventions. We are as a community, we don't have good studies evaluating interventions, and I really have this problem because it's good that my government asked me for advice because they think they should give some recommendations, and we have a number of recommendations, and which go from night workers having to wear dark glasses when they go out of work so as not to get too much light and go to sleep, and yeah, I'm okay with that. That looks reasonable. But then we are asked, is it better to do fast rotation or slow rotation? 2 nights during a week, or just work for 1 month night and then change to. And for the immediate effects, we can do recommendations in terms of sleepiness, for example. For the long-term effects, we just don't have the data, and the little data we have are not. So there are a lot of things, and there are a lot of business here. There's people suggesting glasses that protect you that don't work, and this type of things. So there's a lot of issues like that are very important. And then there are all sorts of etiological research and identification of susceptibles and that stuff. Oh, there's a lot of stuff that can be done at the local level. Yeah. Okay?
I think Molly has one.
Okay.